Hey everyone! So in the last video we talked a little bit about prototrophy and auxotrophy in E. coli or other bacteria, and now we are going to take a look at how Lederberg and Tatum used the auxotrophic mutants to, I guess, discover that bacteria can transfer genetic information from one cell to another, or at least to gather the first evidence that this is possible. So again, sorry about my hands. Did some plumbing work in my house yesterday, and I'm having trouble getting the grime off of my hands. So now Lederberg, again, I'm assuming that's how we say that name. Tatum, I know. Oh, let me get a different pencil here. Tatum in the 1940s. Pencils are breaking. Okay, they are working with two different strains of E. coli. We're going to call it E. coli strain A and E. coli strain B. Now, these are our auxotrophic strains, and they have different auxotrophies or auxotrophic requirements. So E. coli A is mutated in genes required to make methionine and biotin. So E. coli A requires methionine and biotin to be in its medium. So I'm going to put 3 more genes in here. 3+ for threonine plus, Lu plus for leucine plus, and Thi plus. So for all the genes required to make these 2 amino acids and thiamine, the vitamin. So E. coli is wild type for all the genes required to make these guys right here. So they don't need, this strain A does not need these compounds, these 2 amino acids, this vitamin doesn't need those in its medium. E. coli B, however, doesn't require methionine or biotin. It has all the genes needed to make these 2 compounds. But it is mutated in at least one to make threonine, 1 gene to make threonine, at least 1 gene to make leucine, and at least 1 to make thiamine. So these guys were doing an experiment, and what they did was essentially, I think the best way to describe what they did was they took 100 million. Let's assume they didn't do this exactly, but you're going to get the point when I describe it like this. They took 100 million E. coli A cells, and they plated those cells onto minimal medium. And we already talked about minimal medium, and we know minimal medium doesn't include methionine and doesn't include biotin, so we're not too surprised that, you know, they didn't see anything surviving. They plated the cells, let the plates sit for a day in the incubator. Nothing grew. Same thing, so 100 million cells of A. Same thing for 100 million cells of B. Now, I'm just assuming they took these and they plated them directly, no incubation or anything like that. So even though we don't know I don't know the exact details, you should be able to see what's going to happen after this next step. So they plated B on minimal medium, 100 million cells, and let the plate sit out on the benchtop or in an incubator for 24 hours or whatever, and nothing grew. So there were no surviving colonies here. So which makes sense, because E. coli B requires threonine, leucine, and thiamine to be in its environment, and this is minimal medium, doesn't have those, so none of the cells survived. However, when they, let's say, plated 50 million of A with 50 million of B, well, they saw colonies, which was weird, right? And again, so, I'm being kind of exact with the numbers because I'm doing that for a reason. And there's a contrast here, right? So when we plate 100 million cells of A onto here, no survivors, 100 million cells of B onto here, no survivors. But when we mix them and plate the same number of cells, we see, well, lots of colonies. I think it was like 10 or something like that. So the point is, you know, A cannot survive alone, B cannot survive alone, but A and B together, well, something is going on here. And one of the hypotheses was maybe A is transferring DNA from to strain B, or maybe B is transferring DNA to strain A. Another possibility is maybe A is leaking some nutrients that B needs, and B is leaking nutrients A needs. We're going to see why that's not the case in a second. Another possibility, which is discounted by these results on the left and the right here, is that there are some reversion mutations or back mutations that is converting methionine minus to methionine plus and bio minus to biotin plus in strain A. So what does that mean? Let's call this a back mutation. Back mutation. And a back mutation would be like, okay, let's start with E. coli wild type and let's mutate it. Now, we mutate it, and let's say we get E. coli met minus out of there. So some mutation in the gene required to make methionine occurred, and now we have the E. coli met minus strain. Well, let's say we mutate again, or say a mutation happens randomly, and takes that same mutation, whatever happened here, reverts it back. So now it's wild type again, or has the wild type sequence. It no longer requires methionine. I should change this to met plus. So E. coli met plus, mutated, E. coli met minus, mutated again, back to met plus. That's called a back mutation. And how can that happen? Well, let's assume E. coli met has an ATG somewhere in there. This sequence is necessary. This is a DNA sequence in adenine, thymine, and guanine. They say this is important for a function of the protein. We mutate it, and now let's say it's AAA. Okay, well, it no longer functions because it's not ATG. A back mutation would be somehow we mutate these back to ATG. Okay, and it's working again. So that's an example of a back mutation. So Lederberg and Tatum's experiments suggest that back mutations aren't the cause of these colonies, because when we plated 100 million cells of A, we didn't see any survivors, right? And we would have expected at least some of these colonies that showed up here to have also been here if it was the result of back mutation. So something about mixing these two together allowed for production of these colonies. So how do we know it's not like A is leaking nutrients and B is picking up those nutrients, or B is leaking nutrients and A is picking those up? Well, there's another experiment called the YouTube experiment that was done shortly after Lederberg and Tatum's experiment. And what this experiment did was it would put strain A over here and strain B over here, and it had a filter here so the bacteria couldn't touch, but the nutrients could go back and forth. So if strain A was leaking nutrients that B could use, or B was leaking nutrients that A could use, then I should say this is like plugged with a cotton ball or something, and this one is, we have a pump in here that forces the liquid back and forth. So if this was true, then you would expect some of these, the nutrients to allow some of these to grow. And then when bacteria cells were isolated from these and plated on the minimal medium, then, oh, I'm sorry, that's not how I've been diagramming it, right? So minimal medium, so nothing would grow. So nothing. I'm putting X's here instead of colonies. So whether you tested strain A or strain B, nothing would grow here. And so what this told researchers was that the bacteria actually needed to be in contact with one another. So whatever was allowing for this to happen when the bacteria was mixed, the U-tube, this is a U-tube, the U-tube experiment is telling us that for this to happen, the bacteria need to be able to touch one another. So it can't simply be nutrient leaking from A and B picking up the nutrients, or B leaking nutrients that A is picking up. So they have to touch for whatever phenomenon we're seeing here. The bacteria have to be able to touch. And it turns out that we think it's conjugation. We think conjugation is going to, is what allowed Lederberg and Tatum to get these results. Okay, so in the next video, we'll take a look at, actually have a meeting right now, so in the next video, we'll take a look at how conjugation is thought to have allowed this to happen in Lederberg and Tatum's experiment.